Hello, everyone. So today I'm going to talk about Worcester's own Martha Chase. So I can tell you all about Martha Chase, and I will. But I want to start out why I like Martha. For starters, she went to Worcester, and I think it's pretty neat that both she and I went to the same school and studied the same thing. Second, she did the most work without a graduate degree, and that's pretty impressive. And third, for all her work, she isn't really well known, and I think that's pretty sad. So, who is Martha Chase? Martha Chase was born in Cleveland Heights, Ohio, and she graduated from Worcester in 1950. She assisted in Alfred Hershey's experiments, which confirmed that DNA was the factor that transferred information between cells. She worked with various other biologists throughout the 50s, including the phage group. The phage group was a group of researchers who studied genetics and proteins in the 40s and 50s. Afterwards, she went to the University of South Carolina to get her degree in microbiology. Unfortunately, she faced career and personal setbacks, leading her to leave research at the end of the 60s. So what did Martha do? Well, she worked in genetics and biology. What is genetics? Genetics is the study of genetic material and its function and heritability. Heritability being its ability to pass traits to its offspring. But what is genetic material? Genetic material is the parts of an organism that make up the structure and traits an organism and carries those traits through generations. So it's DNA and RNA, and at the times of the 40s and 50s, some scientists thought proteins counted as genetic material. Now, what is biology? Biology is the study of viruses. Viruses were interesting to early geneticists as they were relatively simple and made up of entirely genetic material, or what was thought to be genetic material. The work of scientists like Martha proved DNA was the genetic material and helped create the modern fields of genetics and microbiology. So let's get to the experiment she was famous for, the Hershey-Chase experiment. This experiment was trying to determine if protein or DNA carried genetic material. At the time, most scientists thought proteins carried genetic material as it was much more complex. Chase and Hershey did many experiments to determine if DNA or protein is the genetic material. However, they're most famous for their last experiment. So during their experiment, they decided to put radioactive components into bacteriophages or viruses, and those radioactive components were used to label and identify the DNA and proteins. Phages were labeled with phosphorus 32 for DNA, or alternatively, sulfur 25 for proteins. The phages were then inserted into the bacteria and allowed to infect the bacteria. Bacteria were then put into the blender to separate the phages, the bacterial DNA, and the bacterial proteins from one another. Cells were then checked to see if there were any radioactive components in the cell. They found that in the cells infected with radioactive DNA still had that radioactive DNA inside them. This proved that DNA probably carried the genetic material, and that proteins were not the genetic material, as they were not found inside the viruses. This experiment allowed for more in-depth studies to determine the exact function of DNA in organisms and how it carried out the genetic material. Their work was published in the Independent Functions of Viral Proteins and Nucleic Acid in Growth of Bacteriophage. This work helped win Hershey the Nobel Prize in 1969. Unfortunately, he did not share that prize with Chase. What makes Martha Chase unique is that she gets credit for this paper despite being a lab assistant. This is unusual as assistants don't usually get credit unless they were essential to the development of the experiment. Well, that's it, everyone. I hope you learned something today. I know I have. See you in class.